Physiology of the digestive system. So as a review, digestive system is an organ system that processes food, extracts nutrients, and eliminates residue. There are 5 main stages of digestion: ingestion, the selective intake of food, so bringing food into the body; digestion, the mechanical and chemical breakdown of food into a form that's usable by the body. So mechanical would be physically breaking food down into smaller pieces, while chemical means breaking it down at the chemical level into absorbable monomers or absorbable independent units that we can actually bring into the bloodstream. Absorption, the uptake of nutrient molecules into epithelial cells of the digestive tract and then into the blood and lymph. So the whole reason why we're going to eat or ingest food from the environment is because we need to maintain all of the processes that are going on inside the body. And all of those processes require energy, and some of those processes require some building blocks so that we can build our own tissues and our own cellular components. So through the process of absorption, we're able to take the nutrients that we need from the digestive tract and absorb it into the body. And we absorb it into the bloodstream, or if it's fats, we absorb it into the lymphatic system. Compaction would be absorbing water and consolidating the indigestible residue into feces. And defecation would be the elimination of feces. So what are those major macronutrients or major macromolecules that we care about? So macromolecule is talking about a giant molecule, while a macronutrient is talking about those nutrients that are needed in higher concentrations or higher quantities. So for the macronutrients, even though water isn't a macromolecule, water's tiny, H2O, it's just 1 oxygen, 2 hydrogens. That is, however, considered a macronutrient because we need water in large quantities. Carbohydrates, lipids, and proteins are also macronutrients, so we need those in larger quantities. Carbohydrates, as a review, those are sugars, and sugars consist of building blocks of saccharides. So monosaccharides, mono meaning one, monosaccharides are the independent building blocks of carbohydrates. And we put 2 of them together, we get disaccharides. If we put a bunch of them together, we get polysaccharides. Lipids. Lipids are fats. And there's all different types of lipid molecules that are relevant to the human body. We've got phospholipids, which are part phosphate, part lipid. We've got cholesterol, which is a form of a lipid. And then we've got the main storage and main useful type of fat, and that's called a triglyceride. And a triglyceride has a glycerol group attached to 3 fatty acids, and those fatty acids can change in structure to have more or less hydrogens in them to make them saturated or unsaturated. And then we can chemically modify them to be considered trans fatty acids, and there's different ways that we can combine those, but all of this should be a review from your general E&P course. Then we have proteins. Proteins are just proteins. So like carbohydrates are sugars, lipids are fats, proteins, just proteins. And proteins come in a variety of different forms, and they have a variety of different functions. Some of them are structural, so they just build up the structure, the bulk of the cell. Some of them are functional proteins, and those functional proteins are typically called enzymes. They're going to speed up a reaction. So they reduce the activation energy for a reaction to occur. They make reactions happen a lot faster, and that's how things get done in the body. So we're going to talk a whole bunch of enzymes in the next lecture when we talk about metabolism. But proteins, your building blocks of those are going to be primarily amino acids. And amino acids are going to be put together into a primary, secondary, tertiary, and quaternary structure. If we break these down into the amino acid form, we can easily and readily absorb those into the digestive tract. So inside the digestive tract, our goal is to extract as many of these building blocks as we can and absorb them into the bloodstream or into the lymphatic system, get them to the liver, where the liver is going to process these. and it's either going to store them or convert them, or it's going to use them to build something new. And once that happens, then they get transported to the various locations where they're needed in the body. Now, water doesn't need to be digested. Water can be absorbed directly, and water plays a major role in regulating the pH, fluid balance, the different types of molecules that are present inside the bloodstream. So water plays a major role in overall homeostasis. So a review of the pathway of digestion. So food comes into the mouth, goes to the stomach, or through the esophagus into the stomach, and then we enter into the small intestine, which has three major regions: the duodenum, jejunum, ileum, followed by the large intestine. And the large intestine, also known as the colon, we start with the cecum, which is just the initial portion, and then we have the ascending, transverse, descending, sigmoid colon, rectum, and anus. So that's the pathway that food would travel through the digestive system. There's also these accessory organs. We have the liver, gallbladder, and pancreas that we'll talk about. We'll learn a little bit more about their specific functions in the body. So digestion and absorption. Salivary and gastric enzymes partially digest proteins and lesser amounts of starch and fat in the stomach. Most digestion and nearly all absorption occurs after chyme has passed into the small intestine. So if you remember from general EMP, when we bring food into the body, we break it down in the mouth into smaller pieces. There's some salivary enzymes there, like salivary amylase, which can break down some sugar, salivary lipase, which can break down some lipids. But pretty much the lipids, because fats are hydrophobic, they don't like to be near water. As soon as you take, let's think about salad dressing. If you take a bottle of salad dressing and you mix it up a whole bunch, it's going to be mixed thoroughly throughout, but if you let that salad dressing sit for a while, it's going to separate again. So the fatty layers of the salad dressing are going to separate away from the water layers. It's kind of what happens with salivary lipase. So the lipase is going to break the bonds of lipids in the mouth, but if we let it sit for a while, which we do on the pathway to the stomach, and then while it's in the stomach until it gets into the small intestine, those fat molecules are allowed to come back together. So there's very little amount of actual fat digestion that happens in the salivary juices, but some happens. Then when we get to the stomach, we've got a lot of protein digestion that happens in the stomach, but a lot more will happen in the small intestine. Stomach does not absorb any significant amount of nutrients. It does absorb aspirin and some lipid-soluble drugs. Alcohol is absorbed mainly by the small intestine. Intoxicating effects depend partly on how rapidly the stomach is emptied. So if you ever wonder if you drink on an empty stomach, if you do drink at all, if you drink on an empty stomach, typically absorbed a lot faster, and you feel the effects a lot faster than if you were to drink on a full stomach, because, as it mentions here, it depends partly on how rapidly the stomach is emptied. Regulation of gastric function. So gastric secretion and motility is regulated by nervous and endocrine systems. Increase gastric secretion and motility when food is eaten, and suppresses them when the stomach empties. Gastric activity is divided into three phases: the cephalic phase, which is the stomach being controlled by the brain; the gastric phase, the stomach controls itself; and the intestinal phase, the stomach is controlled by the small intestine. The phases overlap and they can occur simultaneously. So this diagram shows neural and hormonal control of the stomach. So in the cephalic phase, the vagus nerve, which comes from the brain, stimulates gastric secretion even before food is swallowed. So just by bringing food into the body, into the mouth, and starting to move the food around and tasting the food, that vagus nerve will start to stimulate secretion of gastric enzymes. Then we have the gastric phase. When food comes into the stomach, the food physically stretches the stomach and activates the mesenteric and vagovagal reflexes. These reflexes stimulate gastric secretion. Histamine and gastrin also stimulate acid and enzyme secretion. Then we have this intestinal phase. Intestinal gastrin briefly stimulates the stomach, but then secretin, CCK, cholecystokinin is CK, and the enterogastric reflex inhibit gastric secretion and motility, while the duodenum processes chyme already in it. Sympathetic nerve fibers suppress gastric activity, while the vagal or parasympathetic stimulation of the stomach is now inhibited. So basically what happens is if or when the food gets into the duodenum, we want the stomach to stop digesting. Because if the stomach continues to pump out gastric enzymes and there's no food in there, that's when those gastric enzymes and the hydrochloric acid that's released can start to damage either the stomach. Those enzymes can creep up into that lower esophageal sphincter, up into the esophagus, and eventually into the back of the throat, causing heartburn. Those enzymes can leak into the duodenum, causing some intestinal upset there. So we want the stomach to start kind of getting ready to receive food before the food gets there. So that's the cephalic phase. We want the stomach to be really active when food is in there. That's the gastric phase. And when food is leaving the stomach and going into the intestines, we want the stomach to stop being so active. And that's the intestinal phase. So this kind of lays out a little bit more detail about each one of these phases. The cephalic phase, the stomach responds to sight, smell, taste, or thought of food. Sensory and mental inputs converge on the hypothalamus. The hypothalamus relays signals to the medulla oblongata. Vagus nerve fibers from the medulla stimulate the enteric nervous system of the stomach, stimulating gastric secretion. 40% of stomach's acid secretion occurs in the cephalic phase. The gastric phase, it's the period in which swallowed food and semi-digested protein activate gastric activity. 2/3 of gastric secretion and 1/2 of acid secretion occur in this phase. Ingested food stimulates gastric activity in two ways. By stretching the stomach, so it activates short reflex mediated through the mesenteric plexus, activates long reflex mediated through the vagus nerves and the brainstem, and by increasing the pH of its contents. The gastric phase continued. So gastric secretion is stimulated by three chemicals: acetylcholine, which is secreted by the parasympathetic nerve fibers of both reflexes; histamine, a paracrine secretion from the enteroendocrine cells in the gastric glands; and gastrin, a hormone that's produced by enteroendocrine G cells in pyloric glands. The intestinal phase, so the duodenum responds to arriving chyme and moderates gastric activity through hormones and nervous reflexes. The duodenum initially enhances gastric secretion, but soon inhibits it. Stretching of the duodenum extenuates the vagovagal reflex that stimulates the stomach. Peptides and amino acids in chyme stimulate G cells of the duodenum to secrete intestinal gastrin, further stimulating the stomach. Soon, acids and fats trigger the enterogastric reflex. The duodenum sends inhibitory signals to the stomach by way of the enteric nervous system. The duodenum also signals the medulla to inhibit vagal nuclei, reducing vagal stimulation of the stomach, and stimulate the sympathetic nervous neurons, sending inhibitory signals to the stomach. Chyme also stimulates the duodenal enteroendocrine cells to release secretin and cholecystokinin, CCK. These stimulate the pancreas and gallbladder, but suppress gastric secretion. So you should be keeping track, and I do have this laid out in the content guide, but you should be keeping track of what's stimulating what and what's stopping what, and where are these things coming from and what's causing them to be created. Gastrin secretion declines and the pyloric sphincter contracts tightly to limit chyme entering the duodenum. Enteroendocrine cells also secrete glucose-dependent insulinotropic peptide, or GIP, originally called gastrin-inhibiting peptide. It stimulates insulin secretion in preparation for processing nutrients about to be absorbed by the small intestine. So this is a diagram that shows the feedback control of gastrin secretion. You can go ahead and pause the video and review the diagram. So now we have some accessory organs. So food has gone through the stomach, it's gotten into the small intestine. We've had some carbohydrate and lipid digestion, a lot of protein digestion, and now we need to continue the digestion of the rest of the nutrients, so the rest of the sugars, fats, and proteins. So the liver is a reddish-brown gland that's located immediately inferior to the diaphragm. It's the body's largest gland, and it has the ability to regenerate like no other organ in the human body. Huge variety of functions, but only one contributes to the digestive process, and that's the secretion of bile. So here you can see a diagram that outlines the functions of the liver. You can go ahead and pause the video if you'd like to learn about all of the different functions of the liver. Here's what the liver looks like and where it's located in the body. On the underside of the liver is the gallbladder. Microscopic anatomy of the liver. So the hepatic triad consists of a branch of the hepatic portal vein, a branch of the hepatic artery, and a bile duct. So those are all kind of together, and that makes up one hepatic triad. And there's many of these triads in the liver. Hepatocytes. Hepatocytes are liver cells. After a meal, hepatocytes absorb from the blood glucose, amino acids, iron, vitamins, and other nutrients for metabolism or storage. Between meals, hepatocytes break down stored glycogen and release glucose into the blood. They remove and degrade hormones, toxins, bile pigments, and drugs. And they secrete into the blood albumin, lipoproteins, clotting factors, angiotensinogen, and other products. So let's kind of break down all of these different things that the liver is doing. So, hepatocytes, your liver cells, these are the cells that are going to absorb the nutrients from what you've eaten. And it's going to take these nutrients and either store them in the body for later use, or it's going to use them in creating energy or making products that are needed in the cell or in the body. When you're not eating, you still need energy. So let's say you ate a big breakfast, you skip lunch, and then you eat dinner. In that skipping lunch process, it's not like your body can just shut down and not need glucose anymore. Like, you need a steady supply of sugar throughout your day, and that keeps you going. And if you can't use sugar, then you're going to need lipids or proteins in order to make energy from those. So the hepatocytes, or your liver cells, are going to break down stored glycogen. Glycogen is how we store, it's the storage form of sugar. So we take multiple glucose molecules and clump them together into long chains and branches, and those are known as glycogen molecules. Well, those are stored inside of certain liver cells. And then if you need sugar, the hepatocytes themselves just break apart that glycogen to release glucose into the blood. Remove and degrade hormones, toxins, bile pigments, and drugs. So they package up a whole bunch of wastes and things that aren't needed. And secrete into the blood albumin. Albumin is the major blood protein. It allows your blood to maintain colloidal pressure. In other words, the fluid in your bloodstream is there because of albumin. Albumin, and we'll learn a lot more about this when we talk about fluid regulation near the end of the semester, but albumin allows for water to be maintained in the bloodstream and not be lost by passive transport processes. Lipoproteins. Lipids can't just be transferred through the bloodstream. They need to be attached to a protein. because lipids, fats, don't like water. So if you try to mix a whole bunch of fats through the bloodstream, they're going to clump together and they're not going to transport very well through the bloodstream. So in order to transport lipids, and we need to transport lipids, we need lipids all over the body. Every single one of your cells needs lipids, not only to make up the cell membrane, but their lipids are used for a lot of different functions inside the cell. So in order to transport those lipids to where they need to go, we need to first attach a protein to them, and then we can transport them through the bloodstream. Clotting factors, angiotensinogen, and that's going to play a role in regulating fluid balance as well, which we'll learn toward the end of the semester, and other products or processes. So here is kind of the relationship of the gallbladder and pancreas to the duodenum. The gallbladder and bile. So we talked about the liver already, and we said the only function that the liver plays in digestion is making bile. So what happens is you've got red blood cells, and red blood cells do not have a nucleus. They are basically just big discs that are filled up with hemoglobin. And hemoglobin, we'll learn a little bit more of the structure of hemoglobin later on in the semester, and this may even be a review from your general A&P class. But hemoglobin is made up of 4 heme groups. At the center of each heme group is an iron, and at the center of the iron could be an oxygen molecule, and that's how oxygen is transported through the body. Well, when red blood cells get old and they die, they get filtered out in the spleen, and if they're broken, all of those cellular components get released into the bloodstream. Well, now those cellular components from the bloodstream are going to travel to the liver. When they are initially created, it's created as unconjugated bilirubin. So hemoglobin is broken down first into this molecule called biliverdin, which has a greenish color, and then from there it's broken down into bilirubin, and bilirubin has a yellow color. And it's unconjugated bilirubin until it gets to the liver. The liver then conjugates the bilirubin, so it changes the structure of the bilirubin, and it takes that and puts it into the gallbladder. What the gallbladder does is it stores and concentrates the bilirubin. And in addition to the bilirubin, there's other substances that go together to make what's known as bile. It absorbs the water and electrolytes from bile and concentrates it. Eventually, anything that is not secreted in the feces is going to be filtered out of the bloodstream through the kidneys. So bile is a fluid that's secreted by the liver, containing minerals, cholesterol, neutral fats, phospholipids, bile pigments, and bile salts, or bile acids. Pigments and color, the yellow-green color when secreted by the liver becomes intense green when concentrated in the gallbladder. Bilirubin is the primary bile pigment from hemoglobin decomposition. Bacteria in the large intestine metabolize bilirubin to urobilinogen. About half of it's reabsorbed by the small intestine. The kidneys then convert it to urobilin and excrete it in the urine. The rest is converted in the intestine to stercobilin, which is responsible for the brown color of feces. Bile acids, also known as bile salts, are steroids synthesized from cholesterol, and they aid in fat digestion along with lecithin. Bile gets to the gallbladder first by filling the bile duct, then overflowing into the gallbladder. Liver secretes about 500 to 1,000 mils of bile daily. 80% of bile acids are reabsorbed in the ileum and returned to the liver, where hepatocytes reabsorb and resecrete them. The enterohepatic circulation, the route of secretion, reabsorption, and resecretion of bile acids 2 or more times during digestion of an average meal. 20% of bile acids are excreted in the feces, and that's the body's only way of eliminating excess cholesterol. The liver synthesizes new bile acids from cholesterol to replace those lost in feces. Gallstones, so biliary calculi, are hard masses in either the gallbladder or bile ducts. They're composed of cholesterol, calcium carbonate, and bilirubin. Cholelithiasis is the presence of gallstones. It's most common in obese women over 40, but also men and even children. It can occur when cholesterol becomes too concentrated and precipitates as crystals that grow in size. Painful obstruction of ducts results in jaundice or yellowing of the skin, poor fat digestion, and impaired absorption of fat-soluble vitamins. Gallstones are usually removed by laparoscopic surgery. So this is a picture of gallstones. So, funny, random side story that I'm going to tell you real quick. I had a student in the first university that I ever taught for. I was teaching this class many, many years ago. I'm not going to tell you how long ago. It was a while ago. And this student had an excuse for everything. Every day was a different excuse why this student couldn't come to class. So I was giving the lecture on the digestive system. I was talking about gallstones, and then I talked about the intense pain that you can get from having gallstones and how the pain irradiates through to the back. And all of a sudden, she raised her hand and said, I'm having a lot of pain. I think I might have gallstones. So I said, no problem. You can go, and kind of brushed it off like, here's another excuse as to why she can't be in class. Clever excuse, because it was related to the lecture content. At least she was paying attention to the lecture. But again, thought it was just an excuse. About a week later, she returns, and she's got pictures of her surgery. She actually had gallstones, and she actually had her gallbladder removed. And she had pictures, and she showed everyone the scars from the laparoscopic surgery that was needed to remove her gallbladder. So it was pretty crazy. One time, she finally told the truth, and it was all related to the digestive lecture that I was giving. So kind of a funny story. But she was perfectly fine. She even laughed about it. She thought it was like a crazy coincidence that she would figure that out in the middle of my digestive system lecture. But yeah, kind of an interesting story. So back to this whole bile thing. Now, I did mention this briefly, how bile causes or assists in the digestion of fats. So we'll talk a little bit more about this later on in the lecture, but basically fat molecules, you can use lipase, and lipase will break the fats into smaller chunks, but then the fat molecules want to go back together. When you have bile salts present, the bile salts will help emulsify fats, which means once the fats are broken down into smaller pieces, bile salts will surround each one of the smaller fat molecules and prevent it from going back together into a larger fat molecule. So it basically ensures that once it's digested, it stays digested and it stays separated. The pancreas. So pancreas is a flattened, spongy retroperitoneal gland posterior to the greater curvature of the stomach. Both an endocrine and exocrine gland. The endocrine portion, pancreatic islets that secrete insulin and glucagon, concentrated in the tail of the gland. And then the exocrine portion, about 99% of the pancreas that secretes 1,200 to 1,500 mils of pancreatic juice per day. So here's just an overview of the pancreatic function. You can see the gallbladder is under the liver here, and then we have the pancreas over here. The gallbladder and the pancreas both lead into the duodenum. If we break down the cells of the pancreas, we have alpha cells that secrete glucagon, beta cells that secrete insulin, delta cells secrete somatostatin, and acinar and duct cells secrete the pancreatic enzymes that are used in the digestive processes. Insulin decreases your blood glucose. So if you just had a big meal, you're going to produce insulin, and insulin is going to help the glucose get from your digestive tract into your cells. It's going to help it get into the bloodstream. It's going to help it get from the bloodstream into your cells, and it's going to decrease your blood glucose. Glucagon is going to do the opposite of insulin. Glucagon is going to help increase your blood glucose. So if you haven't had a meal in a while, glucagon is going to go to your liver cells and say, hey, we got to break down some glycogen to release glucose into the blood. And that's going to regulate that function. Pancreatic juice is an alkaline mixture, and that's important. So it contains sodium bicarbonate, which is a base. And if you think about where the pancreas is dumping into, It's dumping into the duodenum. So what just came out of the stomach? A big acid mixture. We've got a lot of hydrochloric acid and we activated those enzymes there. Well, that got dumped into the duodenum. And we don't want acid to just float through the rest of your small intestine. We have to neutralize this acid. So the pancreas produces sodium bicarbonate that helps neutralize the acid in the duodenum. And that allows for the pH to be regulated as it goes through the rest of the digestive tract. That's also very important because in order for the digestive enzymes pepsinogen to be activated to pepsin, to be able to break down protein in the stomach, we need a low pH. So pepsinogen is converted to pepsin. Pepsin is active at a low pH. So it's because of hydrochloric acid that enzyme becomes functional and then it's able to do its job in the small intestine. When it leaves the stomach, when it leaves the stomach, we want to stop that. So we want to shut off those enzymes that came from the stomach, and now we're going to activate the enzymes that are in the small intestine. And by doing that, we need a change in pH. And there's very specific things that will regulate the functionality of an enzyme. Temperature is one of them. pH is another one. And then enzyme concentration, substrate concentration. We'll get into a lot of that, regulating enzymes during our metabolism lecture. But in this case, we have this acid environment of the stomach dumping into the small intestine. We got to neutralize that with some sodium bicarbonate. Zymogens. Zymogens are enzymes that are released in an inactive state, and then they're activated by some type of chemical change or some type of other substance. Zymogen example, I just gave one when I talked about pepsinogen being converted to pepsin by hydrochloric acid. So pepsinogen would be an example of a zymogen in the stomach. The acini secrete the enzymes and zymogens, ducts secrete the sodium bicarbonate, and bicarbonate buffers HCl arriving from the stomach. In other words, it neutralizes that hydrochloric acid. The pancreatic zymogens include trypsinogen, chymotrypsinogen, and procarboxypeptidase. Trypsinogen, secreted into the intestinal lumen, is converted to trypsin by enteropeptidase, and that's secreted by the brush border cells of the duodenum. Trypsin is autocatalytic, which means it converts trypsinogen into still more trypsin. Trypsin also converts the other zymogens into chymotrypsin and carboxypeptidase, in addition to its role in dietary protein digestion. So, if you were to draw this out, you would write trypsinogen. And you could put like an arrow to show it being converted to trypsin. And on that arrow, you would write like enteropeptidase, because enteropeptidase is the enzyme that converts trypsinogen to trypsin. Then what trypsin does, and you can draw a loop back to trypsin, it keeps converting trypsinogen into more and more trypsin. So that becomes an autocyclic or autocatalytic. And then you can also draw an arrow out from trypsin and draw it to chymotrypsinogen. Chymotrypsinogen will be converted to chymotrypsin by trypsin. You can draw another relationship where you draw the arrow down from trypsin, and procarboxypeptidase is converted to carboxypeptidase by trypsin as well. So diagramming this out, drawing out these relationships is going to help you be able to explain it when it comes time to taking a test on this, or to producing an essay on this, or some type of summary of the process. Pancreatic juice. So other pancreatic enzymes include pancreatic amylase, which digests starch, which is a big carbohydrate. Pancreatic lipase, which digests fats. Ribonuclease and deoxyribonuclease digest RNA and DNA respectively. Unlike zymogens, these enzymes are not altered after secretion, but only become fully active when exposed to bile or ions in the intestinal lumen. So this shows kind of a schematic drawing of the activation of pancreatic enzymes in the small intestine. Looking at this diagram is great, but through my own experience of trying to figure out physiology and explain physiology to other people, me drawing my own diagrams really helps. And once you physically draw out your own diagram, sometimes when you go to take the test, you remember the exact picture that you drew this from. And you can make those connections all on your own. So I know it's time consuming, but I highly recommend actually drawing pictures and drawing out these processes so that it helps you study. Now, in the homework assignments that are linked to these lectures, there's endless possibilities of how you can portray your notes. Pictures are really useful. So what I recommend is just print them out and draw a whole bunch of pictures of the processes. It's really going to help you when it comes time to taking the tests. So regulation of secretion. 3 stimuli are chiefly responsible for the release of pancreatic juice and bile: acetylcholine, cholecystokinin, and secretin. So acetylcholine from the vagus and enteric nerves stimulates acini to secrete the enzymes during the cephalic phase of gastric control, even before food is swallowed. Enzymes remain in acini ducts until chyme enters the duodenum. Cholecystokinin, CCK, secreted by the mucosa of the duodenum in response to the arrival of fats in the small intestine. It stimulates the pancreatic acini to secrete enzymes. It strongly stimulates the gallbladder. Why? Why would it do that? Well, let's think. If you just ate fats, that means you're going to have to digest the fats. So if you digest them, you're going to need some bile to help keep the fat molecules separated from each other. So CCK is going to stimulate the gallbladder to squirt some bile into the duodenum at the same time that fats are in the duodenum. Because if you have the bile next to the fats and you digest the fats, then the fats are going to stay digested because the bile is going to help emulsify those fats. And then CCK induces contractions of the gallbladder and relaxation of the hepatopancreatic sphincter to discharge bile into the duodenum. 3 stimuli for the release continued: secretin. So release from the duodenum in response to acidic chyme arriving from the stomach. It stimulates the ducts of both liver and pancreas to secrete more sodium bicarbonate. Why? Well, there's acid coming in, so we got to neutralize it. So we're going to neutralize it with a base, and that base is sodium bicarbonate. Raises the pH to the level that's required for activity of the pancreatic and intestinal digestive enzymes. Enzymes are dependent on temperature and pH and the concentration of the enzyme and the concentration of the substrate. But when we talk about the pH, your stomach enzymes require a really low pH, but your pancreatic enzymes require a pretty high pH or a neutral pH, much higher than what you'd find in the stomach is what I'm getting at. So we need to work on bringing up that pH if those enzymes in the small intestine are going to be active. So the small intestine is the site of nearly all chemical digestion and nutrient absorption. It is the longest part of the digestive tract, and small refers to the diameter, not the length. Small intestine is a coiled tube filling most of the abdominal cavity inferior to the stomach and liver, divided into 3 regions: the duodenum, which receives stomach contents, pancreatic juice, and bile. It neutralizes the stomach acid, emulsifies fats, inactivates pepsin due to the elevated pH, and pancreatic enzymes take over chemical digestion. The jejunum, that's where most digestion and nutrient absorption occurs. Ileum, aggregated lymph node nodules, prominent lymphatic nodules in clusters on the side, opposite to the mesenteric attachment. They're visible to the naked eye and become larger near the large intestine. The ileocecal junction is the end of the small intestine where the ileum joins the cecum, and ileal papilla, formed by thickened muscularis of the ileum, protrudes into the cecum like a pair of lips with ileal orifice between them. Regulates the passage of food residue into the large intestine. Villi are projections with tongue to finger-like shapes. They make the mucosa look fuzzy. They're covered with 2 types of epithelial cells: absorptive cells called enterocytes and goblet cells that secrete mucus. The core of the villus is filled with areolar tissue of the lamina propria. It contains an arterial blood capillaries venule and lymphatic capillary called a lacteal. The blood capillaries absorb most nutrients, but the lacteal absorbs most lipids. So this is what this looks like. And you can see the different cells. You should know what a goblet cell does. That's the mucus secreting cell. And the absorptive cell. What's that called? An enterocyte. And that's going to absorb the nutrients. So enterocytes are going to push the nutrients. They're going to take the nutrients from the solution here and absorb it through. And then your blood vessels are going to actually absorb it from there. So it's going to travel through into the blood vessels. And fats are going to be sucked up by the lacteal. Why can't we just suck fats right up into the bloodstream? Well, as I mentioned before, fats don't like water. There's a whole bunch of water in your bloodstream. The only way that you can transport a fat through your bloodstream is if it's attached to a protein. So you have to first attach it to a protein to create a lipoprotein. And the only place in the body that that can really happen is in the liver. So there's some way we have to get the fats from the digestive tract to the liver. And it can't be in the bloodstream. The lymphatic system is the perfect way to do it. So if we suck it into the lymphatic system, we can transport it into this central kind of absorptive hub called the cisterna chyli. And then from there, there's a duct that dumps it directly into the liver, and the hepatocytes will absorb those fats and then process them. They're going to convert them into all different forms. They're going to convert them into lipoproteins if they need to be transported through the blood, and then they're going to get to where they need to go. Additional features, the duodenum has duodenal glands in the submucosa, which secretes an abundance of bicarbonate-rich mucus, neutralizes stomach acid, and shields the mucosa from its erosive effects. Mucus containing signaling molecules that influence immune cells to tolerate food antigens and beneficial bacteria. Beneficial bacteria, that's the good bacteria that's going to coat our intestines and prevent the bad bacteria from overgrowing. If you wipe out the good guys, that leaves space for the bad guys to grow, and that can cause intestinal infections. So we definitely want to make sure that our good bacteria is intact. Those good bacteria are sometimes called probiotics because they're going to do a lot of good things for us and help our intestines, mostly by physically blocking and preventing the overgrowth or adherence of the bad guys. Large population of lymphocytes through the lamina propria and submucosa of the small intestine, and those are going to intercept pathogens before they can invade the bloodstream. So, bacteria love to get inside our bodies. We have white blood cells waiting in the digestive tract, so that if we do eat some bacteria in our food that we're consuming, the white blood cells can hopefully intercept them and kill them before they get into our bodies. Now, this doesn't work with 100% accuracy. Like, we know that we get infections still. But for the most part, these pathogens are going to be intercepted before they can cause damage. Intestinal crypts secrete 1 to 2 liters of intestinal juice per day, secreted especially in response to acid, hypertonic chyme, and distension of the intestine. Has a pH of 7.4 to 7.8, which is a little basic. pH of 7 would be neutral. Contains water, mucus, but little enzyme. Most enzymes that function in the small intestine are found in brush border and pancreatic juice. So now we're going to go through our macronutrients and how they're absorbed. So carbohydrates. These are the most digestible dietary carbohydrate or dietary substance that we consume. So the most digestible dietary carbohydrate is starch. Starch is first digested to oligosaccharides up to 8 glucose residues long, and then they're digested into disaccharide, maltose, and maltose is finally digested to glucose, which is absorbed by the small intestine. Starch digestion begins in the mouth with salivary amylase. Amylase works best at a pH of 6.8 to 7, and that's the pH you'd find in the oral cavity. Amylase is denatured on contact with stomach acid and digested by pepsin. About 50% of dietary starch is digested before it reaches the small intestine. And then pancreatic amylase resumes starch digestion in the intestine. Upon reaching the small intestine, pancreatic amylase quickly converts starch to oligosaccharides and maltose. Brush border enzymes continue carbohydrate digestion. So there's all sorts of different types of brush border enzymes. We've got dextrinase and glucoamylase, which hydrolyzes oligosaccharides, Maltase, which hydrolyzes maltose, a disaccharide. Sucrase and lactase hydrolyze disaccharides, sucrose and lactose respectively. And then monosaccharides are produced by disaccharide hydrolysis, such as glucose, and those are immediately absorbed. And here you can see that pathway. You can go ahead and pause the video and just kind of make note of what's going on in this diagram. So continuing on, plasma membrane of absorptive cells has transport proteins and absorb monosaccharides as soon as brush border enzymes release them. 80% of absorbed sugar is glucose. It's taken up by sodium glucose transport, SGLT proteins. Glucose is transported out of the base of absorptive cell into extracellular fluid, ECF. ECF is extracellular fluid. It is important to write down these abbreviations because if you happen to see them on a test, you need to be able to know what they stand for. So into extracellular fluid by facilitated diffusion. Sugar entering ECF increases its osmolarity and draws water osmotically from the lumen of the intestine through leaky, tight junctions between epithelial cells. Water carries more glucose than other nutrients with it by solvent drag. Absorption of other monosaccharides, SGLT absorbs galactose. Fructose is absorbed by facilitated diffusion by a different carrier protein and converted to glucose within an enterocyte. Once within the enterocyte, glucose, galactose, and any remaining fructose are transported out of the base of the cell by facilitated diffusion, and they're absorbed by blood capillaries in the villus. The hepatic portal system delivers them to the liver. This is a diagram that shows those processes happening. Pause the video and take some time to review these processes. Lactose intolerance, so it's the cessation of lactase production. So lactose passes undigested into the large intestine. It increases the osmolarity of intestinal contents, causes water retention in the colon, and diarrhea. Gas production by bacterial fermentation of the lactose. So if we don't break down the lactose, the bacteria in our colon will, and then it produces gas. It occurs in many people, 90% of Black Americans, 15% of white Americans, 70% or more of Mediterraneans, and nearly all of Asian descent. For most, lactase production ceases or declines to low levels after age 4. Can consume yogurt and cheese since bacteria have broken down the lactose. So proteins, proteases or peptidases, those are enzymes that digest proteins. They're absent in saliva but begin their work in the stomach with an optimum pH of 1.5 to 3.5. That's where that hydrochloric acid in the stomach comes into play. Pepsin hydrolyzes any peptide bond between tyrosine and phenylalanine. Pepsin digests 10 to 15% of dietary protein into shorter peptides and sometimes free amino acids. Protein digestion continues in the small intestine, so pepsin is inactivated when it passes into the duodenum and mixes with alkaline pancreatic juice. Pancreatic enzymes, trypsin and chymotrypsin, take over the process. They hydrolyze polypeptides into even shorter oligopeptides. You can go ahead and pause the video and take a look at this diagram. And you can pause the video and review this diagram. So oligopeptides are taken apart one amino acid at a time by 3 more enzymes. Carboxypeptidase removes amino acids from the COOH end of the chain. Aminopeptidase removes amino acids from the NH2 amine group end. And dipeptidase splits dipeptides in the middle and releases 2 free amino acids. Carboxypeptidase is a pancreatic secretion. Aminopeptidase and dipeptidase are brush border enzymes. Amino acid absorption is similar to that of monosaccharides, so brush border enzymes finish task, producing free amino acids that are absorbed into the intestinal epithelial cells. Their sodium-dependent amino acid cotransporters move amino acids into the epithelial cells. Facilitated diffusion moves amino acids into the bloodstream. Infants absorb proteins by pinocytosis, maternal IgA from breast milk, and release them into the blood by exocytosis. Lipases, or fat digesting enzymes. So lingual lipase is secreted by intrinsic salivary glands of the tongue. Digests small amount of fat while the food is in the mouth, but more active in the stomach. It's joined in the stomach by gastric lipase. About 10 to 15% of lipids are digested before reaching the duodenum. Before digestion in the duodenum, vigorous antral pumping in the stomach's antrum emulsifies the fat, or breaks up the globs. Emulsification droplets are passed into the small intestine and coated with components of the bile, lecithin and bile salts. Fat droplets further broken down into droplets as small as 1 micrometer by agitation of the intestine segmentation. Coating of bile acids, lecithin, keeps droplets from coalescing. In other words, we surround each tiny little goblet of fat by bile salts and lecithin, and that prevents them from coming back together into a larger clump of fat. Large collective surface area for enzymatic action. Pancreatic lipase acts on triglycerides, removes first and third fatty acids from glycerol backbone, but leaves the middle one. The product of lipase action are 2 free fatty acids and a monoglyceride. So here you can see the fat globule. It gets broken down into smaller pieces and surrounded by lecithin and bile acids. Here you can see pancreatic lipase breaking apart lecithin into smaller pieces, and this first and third triglyceride are removed, so you end up with 2 free fatty acids and then a monoglyceride. The absorption of free fatty acids, monoglycerides, and other lipids depends on minute droplets in the bile called micelles. They're made in the liver. They consist of 20 to 40 bile acid molecules aggregated with their hydrophilic side groups facing outward and their hydrophobic steroid rings facing inward. Bile, phospholipids, and cholesterol diffuse into the center of the micelle to form its core. Micelles pass down the bile duct into the duodenum. There, they absorb fat-soluble vitamins, cholesterol, and the free fatty acids and monoglycerides produced by fat digestion. They transport lipids to the surface of the intestinal absorptive cells, and lipids leave the micelles and diffuse through the plasma membrane into the cells. Micelles are reused, picking up another cargo of lipid, transporting them to the absorptive cells. And here you can see that process of the micelles and those being absorbed. So please take a moment to pause this video and review this diagram. So within the intestinal cell, free fatty acids and monoglycerides are transported to the smooth ER, resynthesized into triglycerides. Golgi complex coats these with phospholipids and protein to form chylomicrons. They're packaged into secretory vesicles that migrate to the basal surface of the cell, releasing their contents into the core of the villus. Then they're taken up into the lacteal into lymph. White, fatty intestinal lymph, chyle, flows into larger and larger lymphatic vessels until it enters the bloodstream. So this is the summary of the digestion and absorption of the major organic nutrients. You can pause the video and review these slides. So nucleic acids, nucleases of pancreatic juice hydrolyze DNA and RNA to nucleotides. Nucleases and phosphatases of brush border split them into phosphate ions, ribose, or deoxyribose sugar, and nitrogenous bases. Membrane carriers allow absorption. Vitamins are not digested but absorbed unchanged. Fat-soluble vitamins A, D, E, and K are absorbed with other lipids. If ingested without fat-containing food, they are not absorbed at all, but are passed into feces and wasted. Water-soluble vitamins, B complex and C, absorbed by simple diffusion, and B12 of bound intrinsic factor from the stomach. So if you think about this, that's significant. So this is why we have to have some fat in our diet so that we can still absorb these fat-soluble vitamins. Minerals, electrolytes are absorbed all along the small intestine. Sodium co-transported with sugars and amino acids. Chloride exchange for bicarbonate, reverse chloride, bicarbonate exchange that occurs in the stomach. Potassium is absorbed by simple diffusion. Mineral absorption continued, so we have iron and calcium absorbed as needed. Iron absorption is inhibited by liver hormone hepcidin to prevent overload. Absorptive cells bind ferrous ions and internalize them by active transport. Unable to absorb ferric ions. but stomach acid reduces ferric ions to absorbable ferrous ions. Transferrin is the extracellular protein that transports iron in the blood to bone marrow, muscle, and liver. Calcium is absorbed through the intestine by different mechanisms. Transcellular absorption in the duodenum enters through calcium channels in apical cell membrane, binds to calbindin protein, so concentration gradient will continue to favor calcium influx. Actively transported out of the base of the cell into the bloodstream by calcium ATPase and sodium calcium antiport. Diffusion between epithelial cells in the jejunum and ileum, and most absorbed calcium is from meat and dairy. Dietary fat retards calcium absorption. Parathyroid hormone is secreted in response to a drop in blood calcium, and that stimulates the kidney to synthesize vitamin D from precursors made by the epidermis and liver. Vitamin D affects absorptive cells of the duodenum in 3 ways. It increases the number of calcium channels in apical membrane, increases the amount of calbindin in cytoplasm, and increases the number of calcium ATPase pumps at the basal membrane. Parathyroid hormone then increases the level of calcium in the blood. Water. Digestive system is involved in water balance. Digestive tract receives about 9 liters of water per day. Water is absorbed by osmosis following the absorption of salts and organic nutrients. Diarrhea occurs when large intestine absorbs too little water. Feces passes through too quickly if the intestine is irritated. Feces contain high concentrations of solute, such as lactose. And constipation occurs when fecal movement is slow. Too much water gets reabsorbed and feces becomes hardened. Absorption and motility. So the large intestine takes about 36 to 48 hours to reduce residue of a meal to feces. Most time in the transverse colon does not chemically change the residue, it just reabsorbs water and electrolytes. Feces consists about 75% water and 25% solids. Solids include bacteria, undigested fiber, fat, small amounts of mucus, protein, salts, digestive secretions, and sloughed epithelial cells.